So sorry for, it's gonna go like till 9, and I had a review last night from 8 to 10, and there was just a common like consensus that reviews this late are kind of unholy, and so I'm sorry it's so late, but you know, we'll just deal with it, right? Okay, so how are we feeling about exam 3 in general? Are there?
It seems like there's less material.
Yeah, so I think there is a little bit less material than the other exams.
Just mean it's going to be more in depth, or?
You know, I really don't feel like this exam is that in depth. So, you know, I thought, because the last exam I know, it was really frustrating because a lot of people studied mechanisms and then there were no mechanisms. And I think this exam is a little better that way. Photosynthesis was huge on it, or is huge on it. Just FYI, yeah. So, yeah, I know it's my least favorite area. So, great. But we'll get through it. So are there any just questions up front before we get started? This is the order of what we'll be doing tonight, and we'll just try to cruise through. If anyone has any questions, just feel free to interrupt me. Or if I say something wrong, you know, of course, let me know. So, okay, so we're going to start out with like really basic chemistry. So for those of you that are in chemistry or have had it, I'm sorry, but we just need to like start with the basics for anyone that hasn't. So if you can tell me, if I have lithium right here, who can tell me what these numbers are, what they represent? Skylar.
The top one's protons and shot together.
Good.
And the bottom one's just protons.
Awesome. So what is this called? Yes. Good. And what would this be called? Yeah, awesome. So when we're talking about stability of an atom, what do we usually use to measure stability? Like what is something stable, what does it have? Good. It has a full octet, which usually for most atoms means 8 electrons in that outer shell, right? So that's just another thing that will be good to know. So I'll just write it up because I can't learn anything if I don't see it on a board. Okay, awesome. So, and then the last thing, like with really basic chemistry, covalent bonds, what are they? Sharing electrons, great. So if I have a single covalent bond, how many electrons are in it? 2. What about a double? Or triple? Okay, good. So you all should be good for all that stuff, all the chemistry on the test, okay? So then let's talk a little bit about reduction and oxidation reactions. I think that this, I don't know if you guys have learned this mnemonic, it's what I learned when I took chemistry, oil rig. So oxidation is losing electrons, reduction is gaining electrons. So let's just talk about kind of what's going to happen with each of these. If something is being oxidized, as I said, it means it's losing electrons. What's going to happen to the charge? Awesome. Yeah, it's going to get more positive. What about the potential energy? Good. So let's kind of talk about why that is. Why are electrons associated with potential energy? Yeah. Exactly. Yeah, so there are potential energy within the bonds. Electrons, when a source receives electrons, it's gaining energy. It now has another electron to do work with. So there is, when you gain an electron, you do increase your potential energy. So of course, it's just going to be the opposite. We're increasing an electron, and it means we'll have a more negative charge. Don't mess up the whole gaining an electron means we have a more positive charge. Just like think through it. Because I know a lot of people would be like, oh, gaining, so we have to get positive. Just think about what actually is going on. You're gaining an electron, so it will be more negative. Okay, so the other thing about potential energy is that if you have a lot of negative charges right next to each other, like what will that do to your potential energy?
You're going to have a lot of potential energy because they
Good.
want to leave.
Yeah, right. So an example he used in class is ATP. You have, okay, I don't want to draw it out, but you have your like 3 phosphates right in a row, and they all have negative charges, right? That has a lot of potential energy because these negative charges don't want to be right next to each other. Okay, so does that make sense to everyone? Yeah. Are there any questions? Okay, so let's just kind of go through a few types of oxidation reduction things you can expect to see, like in context of what we've been learning about in class. So if I have NAD plus, or NADP plus, whatever, and it's going to, no, NAD, I don't know what I'm doing. Where is, oh, no. Yeah, here we go. So, if I have NAD plus and I'm going to NADH, is that going to be oxidized or reduced? Reduced. Awesome. The other thing that you could expect to see would be if I'm going from some molecule A to some other molecule B, and in between there's this transition from NAD plus to NADH. So what that means is we are already established that this is being reduced, and if I'm going from here to here, what can you conclude about the oxidation states between these two molecules?
A was oxidized.
Okay. Yeah, so what he said is exactly right. B in this case is more oxidized. Skylar, do you want to explain why?
So it lost 2 electrons so A lost 2 electrons, and that turned into B, and lost 2 electrons to NAD+, which allowed it to make more bonds and attach effectively to it.
Right, exactly. So are there any questions? Did everyone understand that? Could you guys back in the back hear that? Okay. Okay. So pretty much, guys, that's the extent of oxidation and reduction on the exam. It's pretty basic. This isn't a chemistry class, so he's not going to test it at chemistry with. So are there any questions about oxidation reduction? Yeah.
Can you explain that second one again?
Uh-huh. Yeah, so what's happening here, I'm just asking about the relative oxidation states. So, okay, so when we have, let's first talk about what's going on right here. We have NAD plus and it's going to NADH. So my charge is going down, right? So what does that mean? What happened here? Wait, who said lost? You said lost?
B lost electrons.
Yeah, so A did, okay. Yeah. A did. Yeah, so what happened right here though is we gained more electrons to make it NADH. And so for that to happen, since we have A and we lost our electrons, we put them here with NADH, B is now in a more oxidized state. It has less electrons. Yeah?
So we're saying A and B is some other molecule, and NAD plus comes and steals its electrons?
Exactly. Mm-hmm. So I think that's actually kind of an area of confusion. When we're talking about like NAD plus or NAD8 or ATP, GTP, FADH2, whatever, they're not being formed from these molecules. They're these molecules that will either donate electrons or like phosphates, but they're not forming NAD plus. Does that make sense? Okay. Yeah, that's something. Yeah.
One thing that I think can be kind of confusing is when you start talking about reducers and oxidizers.
So the agents.
Yeah, well, you can say that A is a reducer because it reduces NADH, right? Does he have any of that on the test?
It's a reducing agent.
So I can't like directly answer questions
Okay,
like
yes,
that, but
And so then,
this is what you need to know for oxidation reduction. So it's pretty simple, guys.
Is what he said correct though?
Is he saying that this is the reducer? Well, let me think about that.
It's a reducing
So this is more reduced, and it's losing electrons, thus, hold on, sorry, let me think through this. This is being reduced. So this, yes, this would be the reducing agent.
Okay.
Mm-hmm.
So, would you say that A is being oxidized because it's losing?
Yeah, you can say that, because it is losing the electrons. But the question was just like talking about the general states, A compared to B, which one is more oxidized or more reduced? B is more oxidized, that is correct. Because A is losing electrons to NADH, so B has less electrons. Okay. Are we all crystal clear here?
Crystal.
Perfect. Exactly what I want. Okay, so we'll move on. Alrighty, so the next thing that we're going to be talking about is glycolysis. Let's go over big picture. What's the purpose? So when we're talking about glycolysis, citric acid cycle, and electron transport chain, what is the big picture of it? Okay, I think I heard it somewhere. So, we're producing ATP. ATP is how our body runs on energy. ATP or GTP are involved in like every reaction that involves energy in our body. So we really need it. And so glycolysis is just the first step to do that. So where does glycolysis occur?
inside of plasma.
Perfect. Yeah. So locations are important to remember. So unlike plants, we can't create our own glucose. So we intake this through food. And so we'll have a glucose, and in glycolysis, what is our end product?
Pyruvate.
Pyruvate. Great. So how do we create it from just our glucose molecule here?
you have enzymes that will modify it and then break it down into.
Yeah, great. So we have glucose, and an enzyme called PFK will react with glucose and change it into fructose. Or it's like fructose 1,6-bisphosphate. Not terribly important, just know it's fructose. And then what will happen from fructose? Fructose is the interesting to say.
Pyruvate.
Yeah, great. So it turns into pyruvate. So guys, do not forget this. There are 2 molecules of pyruvate, okay? You have to remember this for everything else to work, to get the right numbers for everything. So of course this whole reaction is going to take energy and it's going to produce energy. That's why all reactions in the body occur. So where would our energy sources or changes be here?
So you have your 2 ATP that come in the energy consumption stage, you call it?
Yep.
Investment, that's right. And then in the energy production phase, when you make pyruvate, you have 4 ATP produced and 2 NADH, or 1?
It's 1 per pyruvate, so it's 2 total. let's talk about net. Net gain of energy, we get 2 ATP, right? Because we're putting 2 in, we get 4 out. And so net gain would be 2 and then 2 NADH. Now one thing, glycolysis, like it doesn't produce a ton of energy compared to the citric acid cycle or the electron transport chain. The main thing is we're getting these pyruvate molecules that can then go be made into more energy. Yeah.
Isn't there also pyruvate on that net?
Oh, well, yeah, sure.
And then for the citric acid cycle, isn't it also only producing 2 ATP?
It produces a ton of NADH though, which
Is a lot of energy.
is a ton of energy. So that's another really good point. Where all of this energy from glucose, basically, what is it going to be made into? Where's all that energy going?
Transport chain.
Good. It's going to the electron transport chain, but it's going to the electron transport chain through NADH. So we have all of this glucose, and because of it, because we're constantly changing its energy state, we produce a ton of NADH. And NADH is the real kicker for electron transport chain. It just produces a lot of ATP. So does everyone understand that part? Good. Let me see what else I thought was important. So I guess something that you might want to consider this kind of goes back to the last exam, where if we cut out this enzyme, what's going to happen? It won't work. So what does that mean? If glycolysis doesn't work, right? We don't have any energy, so it would be like an exercise intolerance. We couldn't,
Fall
you
asleep.
know.
You fall asleep and then wake up.
Yeah. We need PFK to work, and if it doesn't work, we don't have energy. We can't exercise. Okay.
Do you need to know what PFK stands for?
No.
What is the 1,6-biphosphate? Bisphosphate.
What does it look like?
No, what is it? Is it?
So yeah. So actually, it's not super important.
Yeah.
But this ATP, when ATP is used, it's formed into ADP, and you transfer one of the phosphates. And so because bis, that means 2, right? And we're using 2 ATP, which means we're extracting 2 phosphate molecules. Those phosphate molecules are going to go onto the glucose to form this fructose 1,6-bisphosphate. So basically, I don't know where I was going with that. What was the question?
I was just wondering what it was describing. So is it just describing the 2 phosphates on the
Yep.
fructose?
The 2 phosphates on the fructose from ATP.
Why is it called bis? B-I-S?
This is 2.
Why is there S? The S?
I don't know. That's just the way they do it in chemistry.
I got a question.
Yeah.
So there's this fancy diagram at the very end of chapter 7 where your fatty acids can go directly into acetyl-CoA. So let's see if glycolysis.
Your fatty acids can go directly into acetyl-CoA, acetyl-glycolysis. What is that called? I learned the mechanism for it once and it's horrible.
I don't know.
And
Can I see it?
proteins can too. But
Yeah.
carbohydrates, as in glucose, has to go through glycolysis, which is our main source of energy.
Right. Honestly, I can't remember a ton about that, and it's not
So
super important.
if glycolysis doesn't happen, it doesn't necessarily mean we're going to die.
Well, yeah. Because we still have energy sources, this shows, we still have energy sources from proteins and fat, but our main energy comes from glycolysis. So did you have something, did I say something incorrect with the bis?
No, I thought this could be something else.
So yeah, are you in OChem right now?
Yeah.
Is that, so biochem, you call it bis. That's where, I don't know if you call it bis in OChem, but anyways. Okay, so the other thing, glycolysis, as we talked about, it's an energy, it requires energy. So if we have abundant levels of ATP and cellular energy already, will we still go through glycolysis? Probably a little bit, but it's going to be slowed, okay? So it is kind of dependent on how much energy we need. Mhm.
So why doesn't it make more if there's more ATP?
It doesn't make more because it's still, if we already have enough ATP, why waste energy on it?
Does it know? Does it have like
Yeah.
nose?
Yeah. Well.
There's a specific enzyme.
We don't need to know. Yeah, it's gonna go and be big.
Yeah. Okay. Then I won't worry about it.
But there's a specific enzyme that ATP and ADP will bind to, right? To stimulate or reduce the amount of glycolysis happening.
Yes. Yeah. Okay. Yep.
I feel like he's gonna ask us, what's the purpose of glycolysis? Is it A, to make 2 ATP, or B, 2 NADH, or C, 2 pyruvate? And like, what's the most important function of glycolysis? What would the answer to that question be?
That's not all of this. It'd be pyruvate. Pyruvate's important. Yeah. Okay. So are we feeling good about glycolysis? I hope we are. Okay. Yeah, it's
Is
good.
NAD plus and just found in the cell beforehand?
Okay, so here is the question that I cannot answer, and neither can the internet. I've tried. It goes back to evolution,
Yeah.
like what came first, the chicken or the egg. I don't know where our
Honestly,
NAD plus
ATP.
came from originally, but
What is NAD plus?
Nicotine
It's-
and dinucleotide.
it's a big nasty molecule. It's huge.
It's pretty-
So I can look it up and draw it up, the structure up on it.
It's made out of adenosine and nicotine
No.
and nitrogen.
Ah,
Yeah, there's not.
there's nitrogen. I honestly do not know the exact
There's a picture
structure.
of it. I'll give you the picture.
Okay, so yeah, I wouldn't worry about that. Okay, are we good with glycolysis? We can move on. Sweet. I'm gonna leave this up just so we can have a big picture on the board. So before, okay, what must happen to pyruvate before we can go into the citric acid cycle?
Let's turn to acetyl-CoA.
Good. It's turned into acetyl-CoA. And what is produced during this process?
CO2.
Bingo.
Is that part still considered glycolysis, or is that?
It's kind of an in-between. It's not really considered. It's more considered citric acid cycle than it is glycolysis, because glycolysis ends with this pyruvate.
Okay.
But so it's kind of in between a little bit. So, but where does it happen?
Yeah, I think
Yeah,
that's
it's
inside.
when you're going into the mitochondrial matrix.
What are you going across the outer membrane?
I don't know the details of it. Yeah, yeah.
Is it,
Well,
is it in the membrane or is it in the mitochondria then?
I thought it was in the mitochondria.
When he taught us this, he said, "This is a debate in biology. We don't know what happens. I think it happens in the membrane." That's what he said.
Okay,
That's what we're doing.
so we're going within the membrane. I love all the debates. in
But
biology.
he said that's not going to be on the test because it's a debate.
Yeah, yeah.
So that would take us
Does it?
Did I write?
Does it supposed to make an NADH when it does that?
Nope. Okay, so we now have acetyl-CoA. I'm only going to draw this out for one acetyl-CoA, but remember it's happening twice. Okay, so we have two. So we have our citric acid cycle. And what are the products of this citric acid cycle? How many FADH2? Okay, what's up? What else is being produced? Okay, and? And? Good. And so remember it's happening twice, so we have to go around twice for each molecule of glucose, right? No, pyruvate, but there are 2 pyruvates. We go around it twice. So in total, we'll have 6 NADH. This is total just for citric acid cycle.
Not including the carbon dioxide that comes off to make it acetyl.
Right here? No. Oh no, actually, so here's the thing. Sometimes you count these 2 carbon dioxide. I've seen it both ways. Sometimes it's 4 carbon dioxide, sometimes it's 6.
And that's the net.
This is just for this part, though. It's not including glycolysis. But yeah, the net. And then finally, we will have 2 FADH2. Okay. So really, I know, like, at least when I was learning the citric acid cycle, like when I was in 120, it seemed so confusing because there were so many intermediates. But this is really all you guys need to know. So it's a lot more simple than I think a lot of people originally thought.
What does the TCA stand for?
Citric acid. Oh.
Tricarboxylic acid.
Yeah, tricarb, whatever this guy was just saying.
Cycle.
It's the Krebs cycle. So Krebs cycle is synonymous with TCA, synonymous with citric acid cycle. Alrighty, so how is this regulated?
By the production of pyruvate.
Yes. So this is a little bit counterintuitive as well because glycolysis is dependent on how much energy the cell has, but actually citric acid cycle is always going on. It's always regulated. So.
What if you don't have any oxygen?
If you don't have any oxygen, okay, well in that case it couldn't be going on because you'd be in fermentation.
So what do you mean by it's always regulated?
It's, under normal conditions, it's not dependent on the amount of ATP already in the body.
Okay, so, but it is dependent on the amount of pyruvate, right?
It would have to be, yes.
So does it always go at the same speed? Or will it speed up or slow down?
It will always go
depending on
the same
how
speed.
much glycolysis?
I'm pretty sure it will always go the same speed.
Is it a lot slower than glycolysis then?
I honestly do not know. Okay, yes.
Okay. Oh, yeah.
Oh, sorry, no. Come right here, yeah, you.
Okay, here in the book it says that in this intermediate step it produces 1 CO2 and 1 NADH as well.
It's saying NADH right here.
Yeah, in the intermediate step.
In the intermediate step, okay. Wait, who said no? Was that to this? To his comment? Okay, well, that's what it says in the book?
Yeah.
Okay.
And it's on the video online as well, that's why we were confused.
Okay, sorry, that was my bad. Yeah.
So, pyruvate is oxidized, but it's turned into acetyl-chloride. Is that why NADH is created?
Pyruvate is oxidized, so it turns into... Yes, that is right. And then back there.
So does this thing, the Krebs cycle TCA, is that the thing, like, there's a lot of ACOA, is it not going to make very much?
There's a lot.
Because, like,
No, it was
there's
still.
too much ATP won't work, or won't really work.
Yeah, so citric acid cycle is not regulated like that. The thing that you need to know is that it's always regulated. Just go with that, yeah.
Okay.
Yeah. If you're looking just at the citric acid cycle, then it's still 6. If you're counting these 2 carbon dioxide, then you will also do 8 NADH.
So
That's a good point, thank you.
So what does regulate the TCA?
I really don't know. So, yeah, enzymes, they regulate everything.
Because I think, like, in the beginning, once there's too much ATP, it, like, binds up in
Yeah.
the sites of it, and then it's like, oh, we aren't gonna work anymore.
Right, and that's what we've seen.
Similar thing probably happened.
Yeah.
Feedback.
So the 6CO2 that's produced, is that including the ones in there?
Yeah, it's
So would
including,
there
well, it's
be-
including the intermediates.
Would there be 8?
8 CO2s? No. No,
Okay,
it's 4
no,
CO2s
I
if you're
see.
just looking at
Yeah,
the
I see.
Okay. Okay. Yeah, sorry guys, I don't know as much like backgrounds about this type of stuff because I don't know. You pretty much learn it in 120 and then you don't use it again until like you learn the mechanism for it, but then it's not really learning the background. So I'm sorry I can't answer all the questions that well. Okay. So let's go over electron transport chain. You guys already heard me. Do you want to go over it? Because you already heard me explain it during that recitation session. So I'll pretty much explain it the same way, but if it would be helpful, repetition is sometimes helpful. All right. Woo! Okay. Let's do it. So, okay, first of all, where is this happening? In the membrane. In what membrane?
Mitochondria.
Awesome.
The inner membrane, right?
Yes. So we'll have another membrane up here.
The citric acid cycle happens where?
It happens in the mitochondrial matrix. So, like, let's pretend I drew it all the way out here. This would be our cytoplasm. This is our intermembrane space. And this is our mitochondrial matrix. Yeah.
The Kooyman cycle, is that like unknown where it's located, or is that in the membrane?
Oh no, Krebs cycle's right here.
Oh, it's in the
Yep. What's going on? Yeah. Okay, so let's kind of talk about what's going on. Where have we left off with the citric acid cycle? What have we just produced a lot of? NADH. Yeah. And so as we talked about earlier, with glucose, most of our energy is going into NADH. And so these NADH have a lot of energy, and they will become NAD plus and have this proton, and the electron will go to complex 1. So I mentioned this in class, but there were a lot of questions of like, why is it going to NAD plus when we're losing this proton as well? Initially, the hydrogen will take both electrons in the covalent bond with it, which is why you are now electron deficient and you have NAD plus. But both of those electrons will go to complex 1 and go through the electron transport chain, so you'll be left with this proton. So if we just think about like in general terms, when we're in chemistry and something is gaining an electron, what does that usually mean about it? Good, it's getting energy, it's probably more electron negative. It initially was more oxidized. It wanted that electron more. So we start here, with NADH, we now have this electron, and the electron, there's a molecule, is it PQ? It should be PQ. It's just Q. It's a ubiquinone, isn't it? Ubiquinone, that's right. And that will transfer this electron from complex 1 to complex 3. So if ubiquinone is moving about in our membrane, what characteristics can you conclude about it? Yeah. So you can probably conclude that it's nonpolar. Something that's nonpolar is usually hydrophobic. It likes lipids. It also can't be stationary, right? It must be mobile if it's moving our electron. So. from complex 1 to our more oxidized complex 3, and it will take, the electron will then go to complex 4, where it will go to cytochrome C.
Oh, it's gonna go to
It's going to go to oxygen, right? Yeah, so we have our oxygen here. Now oxygen is the ultimate electron acceptor in this whole process. So oxygen will receive an electron. These protons that we're generating and that are just kind of coming through by the whole electron transport chain, it will combine with that and will form water. So if we were to, like, I think he gave an example of this in class, if I remember right. If you breathe in radioactive oxygen, where will you, like, see that radioactivity in your body? In water, because that's what it's going to be made into. So let's see what else. As the electrons are going across, you're pumping up hydrogens into this inner membrane space. And so what you end up doing is you create this wonderful gradient here. And what do we know about gradients? Which way do they want to follow? Where do they want to go? Yeah, they want to go where there's not a lot of other hydrogens. And so it will go down through this molecule called ATP synthase. And when that happens, we get hydrogen down here, which is great because then it can be recycled and go like back through here, right? But what happens with ATP synthase? What does it do? Right. So you talked about this in class. There was like, you remember watching that video, and there were those beta units that would spin. So it spins, and that facilitates an ADP to combine with a phosphate to make ATP. And here is our energy we want. So, one molecule of NADH makes 3 ATP, and one molecule of FADH2 makes 2 ATP.
Could you write that up?
Mm-hmm. So the FADH2 will enter right here at complex 2. And that's because it doesn't have like enough potential energy to enter here at complex 1. And I can always remember this 3-2-B because if you look at what's going on, NADH will go through complex 1, which will pump a proton across. 3 and 4, then they will all pump protons across. So 3 hydrogens, but FADH2, since it has to enter later in the chain, and it's this it's not a channel, it can't pump a proton across. So it can only do 2.
So how does NADH pump 3 hydrogens across if it only has, doesn't it only have 1 hydrogen atom?
Mm-hmm. Yeah, that's a great question. So there were other hydrogens, just by nature of the gradient, that are down here anyways. Not as many, but there are still some going on, and that's how that works.
So is it NADH being oxidized, is that where the electron comes from for the first protein?
NADH being oxidized, yep.
Isn't there another transporter between 3 and 4?
Cytochrome C. Yeah. It's not super important. But, yeah. I honestly can't remember. I'm sorry. This is a fun game. Just ask me all the questions I don't know. So it's on this.
Does it
I figured we
just
should come up with a safe word or something. So
does it just transport electrons?
Yes. Yeah. Okay, it does not matter what side it's on, guys. Cytochrome C is going to act like ubiquinone, or it's, I don't even know if it's called cytochrome C. It's going to act like ubiquinone. It's going to transport this electron from 3 to 4. Yeah. So, okay, so Elise.
I remember representing like as we go down the electron inflection, is each complex more electronegative?
So yeah, so each complex will be in a more oxidized state as you go across.
Meaning they have
Means it's more electron deficient, so it will want that electron more. Yeah.
So what's the total net gain at this point? Or like,
Sorry, what?
what's the total net gain for the electron transport?
Right. So, you know, there's really, that is another thing in science that's up for debate. There's not an exact answer. It's between like 36 and 38, but it's total, yeah, for all of this. But it's not something since it's up for, like, since it's a controversial topic, it's
It's not even known like this.
it's between 34 and 38, is that what it is?
yeah, but I think that might be like total.
Yeah, total.
It's not net. No.
That's from the whole oxidative phosphorus.
No. Yeah.
Why did you go through why NADH produces 3 and FADH produces 2? Yeah.
Yeah. So this is just how I remember it. I don't know if this is right scientifically, but this is how you remember it. So NADH has more potential energy, and it will give up its electrons to complex 1. Now complex 1 is a transmembrane protein, and it can pump an electron across. Then it will go to complex 3, where it can pump I said electron, proton. It can pump a proton across. Goes to complex 3, pumps another proton. Complex 4 pumps another proton. 3 protons total. However, yes, for 3 ATP, because 3 hydrogens can now come down and generate this. However, FADH2 does not have as much potential energy, and so it needs to start here at complex 1, likely because FADH2 is more oxidized than complex 1, so it isn't going to give its electron to that, but it will give it to 2, and then from there it can be passed on.
Yeah. It's complex 2 FADH2 produces 2 ATP. That's all it takes.
Oh yeah. Yeah, totally. So yeah, just 2 going across.
So it's just one electron change the function. Is it a pair or
It'd be 1 electron at a time.
one electron?
Okay, let me look at what else I wanted. So are there any questions about this other ones?
Just out of curiosity,
Uh-huh.
so, like, is up for debate 34 to 38 ATP for like 1 molecule of glucose?
Yep. Yeah. So this this is something that I swear whenever you go with mitochondria is always tested. It's the MCAT's favorite question, I think. If I poke a hole in my membrane, what's going to happen? Good. So if I poke a little hole, all of a sudden before all of the hydrogen would go down through this ATP synthase, right? However, if I poke a little hole, the hydrogens can now go through the hole in the membrane, and we won't get as much of an ATP production because it can go through this as well, and it will destroy the gradient. So this in class, I think it was covered under like the chemiosmotic unit, right? Can someone just tell me briefly what chemiosmotic means? What it was describing? Yeah.
Utilizing chemicals crossing the membrane to create energy.
Good. So yeah, so what he said is exactly right. It's basically an ion gradient, ions moving down the mitochondria. The membrane to make this gradient which happens right here. This is the proton motive force, and we're generating ATP from it.
So the proton motor force is an example of chemiosmosis?
Well, it's kind of just an integral part of chemiosmosis.
Could you use the word substrate level phosphorylation in oxidative phosphorylation to
You guys,
describe this process?
I am so sorry for the mess that was recitation on Friday. Like, I felt so horrible because Matt explained it wrong, and then Preston explained it wrong, and then I explained it wrong, and I'm sure you guys were so confused. And it was, I think, the worst thing I've done as a TA ever. Anyways.
We get it wrong on the test, we just blame it on you.
Yeah, I seriously, I felt so bad. So yeah, so let's go over that. Okay, so oxidative phosphorylation is using glucose to end up with ATP from the electron transport chain. So when we're talking about oxidative phosphorylation, it includes glycolysis, it will include citric acid cycle, and it will include the electron transport chain. Now it gets confusing because the definition of substrate level phosphorylation is that it's mediated by enzymes. And so this is where the confusion in class came in, because glucose requires enzymes to be made into pyruvate, and pyruvate requires enzymes to just carry out the whole TCA. But what you have to look at it, the way you have to look at it is big picture. Where is my ATP coming from? If it's coming from the electron transport chain, then all three processes will be considered oxidative phosphorylation. In fermentation, we're making NAD+, and just like we'll cover this in fermentation, and just the ATP from glycolysis right here. And because that's never involved with the electron transport chain, that's when it's considered substrate level. And we can talk about that more when we get to fermentation. But does that make hopefully more sense than it made on Friday?
So whenever you're referring to substrate level phosphorylation, we can just assume that he's talking about fermentation?
Yeah, so glycolysis fermentation. Yeah, it's mediated by enzymes. That's an important thing to know.
And then everything as a whole is oxidative phosphorylation. The glycolysis, the glycolysis.
Yeah. If glycolysis, if what's going on when I start out with glycolysis results in ATP production from the electron transport chain, it is considered oxidative level phosphorylation.
So substrate level phosphorylation happens in both glycolysis and the citric acid cycle. But oxidative phosphorylation happens in the electron transport chain, but because one doesn't happen without the other, it's considered that all three processes are oxidative.
Yes, but I would not remember it like that.
Great. How would you?
If you are, so just like I said, because you don't want to be confused thinking like, oh, this really is substrate level, but in the whole process it's oxidative. I would just keep it simple. Look at where your ATP comes from. If it's from electron transport chain, oxidative. If it's from fermentation and glycolysis, it's substrate level. Does that make sense, or is that confusing?
Yeah, that's right.
So glycolysis can be both then?
Yes. So.
But what you look at isn't the glycolysis. You're looking at what is my end result. If your end result is the electron transport chain, then it's oxidative. And if your end result is fermentation, then it's substrate.
Bingo.
So what did you say the definition is?
It is mediated by enzymes to produce ATP.
If it makes you feel any better, YouTube doesn't explain it very well either.
You know, I found that too. The internet is horrible at explaining this topic. Because I remember I was looking it up for one of my classes a few semesters ago, and everywhere has like a different definition of it. So, anyways, I'm still sorry about that horribleness that happened. Okay. So I guess really the last thing to cover with like this whole oxidative phosphorylation thing would be the experiment. And this experiment I think is like one of the better ones we've learned all year. So let's quickly talk about what's happening here.
very beginning. J.
J8.
A. Yeah.
I wonder where that's from. I don't know if it would be German. Yeah, I feel like it might be. Oh yeah. Okay. So what happened? How does someone want to explain this, or do you guys want me to explain it? It's all me. Okay. Alrighty. Okay. So what they did is they're like, okay, well, what is producing ATP? Why do we have chloroplasts will produce it and mitochondria produce it, but why? Like, what's happening? And so they took spinach and they isolated the chloroplast from it, and they assumed that there was something going on with the hydrogen gradient. So they decreased the acidity to a pH of 4. Now, one thing to know, like if you haven't had chemistry, when we decrease our pH, we increase our acidity. If we increase our acidity, it means we have more hydrogens. So up here, this has a much lower pH. It's much more acidic than down here. So we had a pH of 4 right here, and we have a pH of 4 right here. Will ATP be produced? No. This won't happen because there's no gradient. There will be no hydrogens moving down because it's not energetically favorable. So my ATP synthase won't be working. So then what they did, and remember that the mitochondria or the chloroplast they used, it has ATP synthase in it, and we'll talk about that in photosynthesis. But then they used another chloroplast vesicle, and it had a pH of 4 again on the interior, but the exterior was a pH of 8. What does that mean? Yeah, so there's less hydrogen. We have a ton in here, and not very much out here. And so now we have this gradient. The hydrogen will move across, and we will have ADP go to ATP. So I'm sorry, I should have mentioned as well, in this extracellular fluid, or it wouldn't be cellular, extravesicular fluid, there would be ADP and an inorganic phosphate. So what was the whole thing? What was their question with this whole experiment?
What's making ATP? Why is ATP?
Yep. And why did they find that it was being made? Good. So remember, this experiment, the same outcome would have happened if it occurred in mitochondria as well, because there's still that same gradient principle, the same ATP synthase principle.
Well, but their experiment wouldn't have worked because the ATP synthase is split, right?
Yes. So sorry, I misspoke. The principle is the same though.
Yes.
And that's important to know.
Okay.
So they would have had to change the acidity. Like they would have had to make it more acidic out here than here. But yeah, but it's the same principle.
Then they also found out that it was electrochemical gradient driving this creation of ATP and not sunlight.
Yeah, it's all about the select.
This happened in light and dark.
Okie dokie. Well, so are we good with all this? No, we're not good. Yeah.
Well, this is a quick question. So in the book, there's another experiment that talks about. Sorry.
Yeah, no.
This one, it's got two different guys, Walter, Ace or whatever. They did a different experiment to figure out that ATP synthesis or ATP synthase did this. So.
Okay.
I don't know which came first or which one's more important.
This one's more important. Okay.
All right.
Okay.
The one that, do you remember the one that he talked about in class where it had the tail on it that was fluorescent, where they saw it spinning? What were the names of those people? Do you know?
No.
Okay.
She doesn't know which one those
So, all right. let's get into fermentation, and this goes back to substrate level phosphorylation. So remember, mediated by enzymes. What's going on here? Well, so we intake glucose, right? And we know that glucose ends up producing pyruvate. And our net gain is 2 ATP. But sometimes we'll use fermentation instead. Why would we use fermentation? Yeah, and there's lack of oxygen. The other time that we could use it, I shouldn't have erased it. So when we were talking about our electron transport chain, what would happen? Okay, let's try to remember what was up there a minute ago. If our last electron acceptor wouldn't accept electrons, what would happen? It would get backed up, and the whole thing would stop. And if that happened, we'd need to go into fermentation because we can't produce ATP based on the electron transport chain if it's backed up. Mm-hmm.
Is that why you're showing 2 ATP produced?
Yeah, so let me explain this. 2 ADP are going in, and it's producing 2 ATP. So this would be net for all glycolysis. So it accounts for the 2 going in and the 4 coming out. So yes, that's why we would use fermentation if we don't have oxygen or if our electron transport chain is backed up. And so we have pyruvate, and the main thing to know is that it's going to go into either what, if we're talking about humans, what will it go into? And if we're talking about like yeast, it will go into ethanol. In the process, it will generate 2 NAD+. Now this is important because if we remember what was going on in glycolysis a minute ago, when we have glucose and we go to pyruvate, we decided that it spit out 2 NADH, right? But as we talked about, NAD+ it's not like something coming off of glucose, it's its own molecule already, and so it's produced from pyruvate in fermentation. So this NAD+ is necessary to produce this NADH in glycolysis. And that's the whole purpose of fermentation, this NAD+. So, and it will end up producing NADH, and this whole cycle kind of goes. Let's see what else. So again, I talked about this already, but it's substrate level phosphorylation, right? And this lactate, this is just kind of a side note, the lactate, lactic acid, when do you have lactic acid build up? It's after you exercise, right? When you get sore, it's because of that lactic acid buildup.
Isn't that just hydrogen attached to lactate?
Yeah. So that's really it for fermentation. Remember, guys, humans do not produce ethanol. We do not make alcohol.
Could you explain one more time where the NADH and NAD+ are coming from? What's happening there?
So where they're coming from initially, because I talked that week.
That's just, they're there. The NADH is already there, the NAD+.
The NAD, what comes first, chicken or the egg? I don't know. But just know that when this, because these could be going on simultaneously. So it's not like while we're producing NAD+ from fermentation, and that allows glucose, which is going on to make pyruvate. That's where that NAD+ can come from. I don't know where it came from initially. I don't think anyone knows that. Yeah?
Is it that the pyruvate is combining with the NADH in order to produce lactate and NAD+?
No, it wouldn't. I don't think that would be it. So, wait, sorry, can you repeat that one more time?
So where does the NADH go?
The NADH? So the NADH, I think it will just be back here and be recycled.
So that's.
But I'm not
what-
quite sure, honestly.
Is that what's producing the NAD plus? Is the combination of the pyruvate with the NAD?
So pyruvate isn't turned into NADH.
Yeah, but is it combined in order to make lactate and NADH plus or NAD plus?
Are NADH and pyruvate combined?
Yeah.
No.
Okay.
No, this is just a product. There are many enzymes going on here to make lactate.
Okay.
NADH or NADH going in NAD plus is a product of that. It's never combining with it. Hold on, sorry.
So just grab the H off the NAD, right? And the NADH, the
Yep.
pyruvate, it just sucks it off. And then it
Yeah.
immigrates to the plus. Right?
So
And it goes
it
into lactate, right?
Kind of. It takes the electrons as well.
Yeah,
Yeah.
that too.
I was just saying to help that, it takes the electrons from NADH, which allows pyruvate to add more hydrogen to it and helps make it lactate. And there's also supposed to be 2H plus with that NAD plus.
Sorry, there's 2H plus going right here.
Right? And with the NAD plus.
Yes.
Right.
What?
In the cycle right there, it's 2 NAD plus plus 2H plus.
Okay.
Right. There you go.
Thank you.
Where in the cell does fermentation occur?
Sodium cytoplasm.
The H pluses are donated by NADH. Do they become a part of lactic acid cycle, or do they just get recycled
I
back?
think they're recycled.
So, and that's where in the future NAD plus grabs the hydrogen to become NADH or recycled.
Sorry, can you repeat that word again?
And so when glucose is oxidized and NAD plus becomes NADH, where does that go?
Where does hydrogen? So it could be. It would just be somewhere in the extracellular fluid, like nearby.
And so that was leading to another question is, if the H pluses don't go to lactic acid, are they then recycled?
Yeah. Okay. All right, so that's pretty much it for fermentation. Are we feeling okay about it?
So, the purpose of fermentation is to change NADH into NAD plus,
Yeah.
it produces glucose, it produces lactate in humans, which is those lactic acid.
Which makes for muscle.
Yeah. Is there anything else that we?
Yeah, so just to reiterate, purpose of fermentation is NAD plus. NAD plus allows glycolysis to occur. Huh? Why does it need it? Because it needs somewhere with the electron. Yeah. Okay, so I'll erase this if that's okay. Okay, photosynthesis. Huh. Okay. Uh-huh.
Does each pyruvate grab two off, or is it two pyruvates?
What you just wrote, is that for one pyruvate or is that for
One pyruvate. No. Wait.
Two.
I think it's two, you're right. Yeah, I'm sorry. Two pyruvate.
You get one NADH or one NAD plus.
I think it would be for both pyruvates. I'm not positive. I'm so sorry, guys. I wish I was good at. I was an expert at this. I'm not. Okay. So photosynthesis, my least favorite unit in this class. But then it's over, so you guys are almost done. So okay, purpose of photosynthesis. What's going on here?
Starch for the energy of the plant, and then also for the structure and I don't know how that was called.
Okay. Yeah, got it. So we have carbon dioxide, we have water, and we have light. That's what's being put in when photosynthesis, and it spits out glucose and oxygen, which is awesome. So with photosynthesis, there are two things going on. There's the light cycle and there's the dark cycle. And we'll talk about what each of these mean. But both happen in the chloroplast. Okay, so I'll talk about that at the end. Let's go through. So let's go over the light cycle first. With the light cycle, we have, I'm just going to draw it out here, what we have going on. Okay, so this is what's going on in the thylakoid of the chloroplast. So we start out right here with photosystem 2. And photosystem 2 will absorb light at 680 nanometers. So what happens is we have this light coming in. Okay, sorry, let me back up. What is in the thylakoid? Like, what is it made of? Yeah, it's a membrane, but that membrane contains pigment, okay? The pigment is what gives it, what reflects the green color. And so remember that the pigment in these photosystem, that's what's absorbing the light. And that's where the whole electron excitation is happening. So what will happen, light will come in, and one of the pigment molecules will absorb it, and it will elevate that electron to a higher energy state, okay? So now we have this electron, and it has tons of energy, but we can't just, this photosystem doesn't just want to lose an electron, so what will it do to compensate? All right, well, I think I heard it. So we'll take water, and it will produce 1/2, this isn't super important, just know that the electron that's being used to replace the electron that's leaving comes from water. That's the source that drives this whole thing. So the electron will go to QB, or QB will transfer the electron from P680 to cytochrome C right here. And as we talked about before, what would be the characteristics of QB? Mobile and hydrophobic, right? It's moving. So it transfers the electron to cytochrome C. Where else have we seen cytochrome C? Electron transport chain. So what's happening is we are pumping the hydrogen down here. The energy that is used with this electron gaining and losing is used to pump a hydrogen across. And it creates this gradient. So next the electron will be transferred via PC to P700. What happens when it reaches P700? It gets excited again. Now this needs to happen because each time the electron is being transferred, it takes energy. And then right here, when we're pumping this hydrogen across, that takes energy. So by the time we get to P700, we're kind of out of energy. So we need more light. So light in P700 will be absorbed at 700 nanometers, and this is photosystem 1. And now our electron has energy again, and it can move to NADP plus reductase. What will happen here? which is cool. But the other thing that's really cool is we have this hydrogen gradient right now. And so we can use our good guy from the electron transport chain, the ATP synthase. We can pump the hydrogen across down its gradient, and in the process, make our ATP. Okay?
So, just a question.
Mm-hmm.
This is how they have it laid out in the book, where you have the NADP tryptase on the end there, and then you've got kind of a corner, and it goes down to the ATP synthase. Is that because we know it actually looks that way, or is it just kind of illustrative?
I don't know. I assume if that's how they are illustrating it, that's probably how it would be, but I don't know. I'm sorry. Okay, yeah.
So in cytosome C, is the electron like kind of dead there? Does it get more excited there, or is it that just in the
So the only times the electron will be excited is when it gains energy. We'll gain energy through light, right? Light is like photons, little energy packets. So it's only, it still has energy at cytochrome C. It runs out at P700. Mm-hmm.
We need to know like the names of the intermediate.
Like cytochrome C?
Yeah. Like if we know what they do,
I would know the names.
So chloroplasts and mitochondria are opposites as to where their hydrogen gradients occur.
Yep.
Right? So this one, the hydrogen gradient is on the inside,
Mm-hmm.
and then mitochondria are just on the inner membranes.
Mm-hmm.
So just with the ATP synthase, it's also in like the membrane, right?
Yeah.
It's not just with that.
Yeah, sorry. So
No, you're good. I'm just
yeah.
Okay, it's not like it keeps going in. It's like
Yeah, it's still in the membrane. Mm-hmm. Okay, sorry. Back here. No, you. That's from you.
This is all happening in the membrane of the thylakoid.
Uh-huh.
And you're just pumping ATP into
Yep. Okay, yeah.
Yes, the Z reaction, or the Z machine,
Yep.
I guess it's the same thing, but just shows like the energy of the electrons, is that right?
Yeah, exactly. So the Z reaction thing that we talked about in class, what it's showing, this electron has the most energy right here, and as it gets transferred, it decreases energy, until we get right here where it's low again, which makes like part of the Z, right? And then it will be elevated in energy, and then it will decrease again. So it's really that light that comes in, that's what excites the electron. So it forms Z. Yes?
So how does it work? Is it the end of NAD plus 2 H plus? But why do you have 2?
Wait, sorry, I don't understand your question.
At the very end where it's making NADPH,
Uh-huh.
you have right there NAD plus 2 H plus?
Yeah, I don't know. That's just the way that they write it. I don't exactly know why. Does anyone else know why that would be?
I might be mistaken, but I think that the way like 2H plus NAD plus, so it's NADPH plus H plus.
Oh, plus H plus? Okay.
Maybe
Thank you.
it's like part of the process.
Can you explain what primary
I don't really know. Yep. Yeah.
What did you say was the overall purpose of photosynthesis?
So the overall purpose of photosynthesis is to produce glucose.
So this is just half of the purpose.
This is just half photosynthesis. This is the light reaction. However, never mind. We'll get into that in a second. Okay, let me make sure I didn't miss anything with what I want to talk about for light. Oh, okay, there actually were a few more things. So why do we see green? Okay. Yeah, so I'm sure you guys all said it. It's because we can't absorb, plants can't absorb that that wavelength of green. And so why would, he talked about this in class, why would trees change color in the fall?
Because the pigments all die, and so they're not doing photosynthesis anymore, and so it's reflecting those lights.
Okay.
But there is still carotenoids, and those do a little bit of synthesis on the green, so it's still reflecting green light, right? But it's the same amount that it was before. But now it's also reflecting a lot of orange and red, and so we see that instead. Is that right?
Mostly. So what happens when we see green, there are also, so we see green because that's what's not being absorbed, right? It's being reflected. However, there's also red and orange and yellow that are not being absorbed, certain wavelengths of red, orange, and yellow not being absorbed. But there's so much pigment and chlorophyll that reflects green that it overpowers that red, orange, and yellow. So when it turns fall and there's not as much sunlight, photosynthesis is kind of a wasteful process because we don't have the light to create glucose. And so the chlorophyll will, it will die a little bit. And as Jeremy said, the other pigments that were in chloroplast will take over, and those will be expressed more. So you can see, now that you don't have that high expression of green, you can see the red, you can see the orange, you can see the yellow.
So just to clarify then, the green light, instead of being reflected as green light, it'll just be reflected as normal light then.
Yeah.
Because
it shouldn't be reflected at all, it should be absorbed, right? Because it's reflected because it's not absorbed. So if we're no longer seeing green light, it would be because we're absorbing it.
Well,
no, is it?
Because the pigments to absorb it aren't there anyway.
But there are other pigments that can absorb that light.
Okay. But it's just not going into photosynthesis.
Yeah, it's not going into photosynthesis.
The light that would have been separated into green light.
Mm-hmm. Let's see, what else? So we've been talking about carotenoids. What is the purpose of those?
Don't they take away the free radicals?
Right. So free radicals are just like electrons hanging out, but they're really dangerous because they're highly reactive and they can go mess with a bunch of other processes going on. And so carotenoids are important because they will absorb those free radicals, and in doing so, they protect the chloroplast. Okay? Or they protect the chlorophyll. Okay, what else do we want to talk about?
Question.
Yeah.
Are carotenoids, are they part of photosynthesis? Like, can they be in the photosystems?
Can they be? I don't think they're in the photosystems, but they're in the thylakoid membrane.
So are they part of photosynthesis?
Do they absorb the? Well, yeah, they can still. So they don't produce free radicals, if that's your question. They can still absorb light and still reflect light. That's actually where part of the color comes from with the red and the orange. But they aren't. They're not going to produce electrons to go down this chain. Only chlorophyll. So okay, so light ends up as NADPH, right? That's where so all the energy that we put in with our photons from light, it ends up making NADPH.
Yeah. Can you explain the cyclic electron, whatever it is?
I don't know what it is. If you can pull the slide, I don't know the name of it.
I know what that is. It's just right here, cyclic photophosphorylation.
Oh, we're good.
It's like.
Yeah, I don't know that. Does someone else want to explain it? Oh, this is such a fun game.
It's basically when there's too much NADPH in the chlorophyll, then the cell will stop it'll stop sending the electrons all the way in this chain, and you'll start cycling them in a circle. So it doesn't take any more water, and it doesn't use the photosystem 2 at all. It just uses photosystem 1 and cycles them down to the QB, and then right back to photosystem 1. The electrons just go in a circle. And then they're still making that gradient of H pluses on the inside, so you're still producing ATP, but you're not making any NADPH because the electrons are not going to NADP plus reductase. They're just cycling in a circle.
Sweet. Okay. do we have that? Good. Okay. So we've talked about this. I just want to make sure that we understand this really well. So absorption, what does that depend on? When something's absorbed, why is it being absorbed? Right. It's at the correct wavelength to be absorbed, and if it's not at that correct wavelength, it will be reflected, right? So green isn't the correct wavelength to be absorbed, therefore it is reflected. What absorbs it?
Yeah.
The pigments in the thyroid. Yeah, cool.
So then, would the P680 be lower than the Does that make any sense?
The P680 be lower?
Because it's like accepting the light, but it's reflecting green. Is that like the correct connection, or like confusing?
I don't really understand what you're saying.
Like, okay, so what?
Is it what you emailed me about? Is that what you're asking?
Yeah, but I'll just ask it after.
Okay, well, because I can answer that. I emailed you back, but it was right before this, so I don't know if you saw. Yeah, so P680, I think, so her email was about, do you mind if I tell her?
No.
Okay, so she was asking, does P680 make electrons go to a higher energy level because it's absorbing at a lower wavelength, which means higher energy, right? And so then in relation to P700, and the answer to that is that P680, it's not, the electrons are not being excited to 680 nanometer wavelength, the corresponding, I'm explaining this so poorly. Okay, let's go back. So when we're talking about energy, this is our equation for it. We have frequency, but frequency and wavelength are inversely proportional. So as we, so she was saying, since this has a smaller wavelength, it should have more energy. And so would it excite these electrons to a higher energy? And I looked online, and I couldn't see anything that like supported or refuted it, but the logic to me makes sense with that, and so I'd assume they would be at a higher energy level. But that energy level isn't going to be the corresponding like 680 nanometers. Okay.
We asked him about that in class.
Oh, you did?
He said that P in the PS1, it is excited to a higher energy level. And he said it didn't, the way he explained it was because of the way that he basically gave a blanket, oh, it's because of this. He didn't want to go into the details.
Okay.
But he said it's just the conformation, the way that they're conformed. This can transfer the energy into the electrons more efficiently or something. So they go to a higher energy level.
Yeah. Great, that's why I was confused, because it makes sense that it would be the other way.
Okay.
But he said that, but...
Yeah, so I couldn't find anything online. I looked and...
I can ask him.
Yeah, I'm sorry. Okay, so sorry, we're almost done with the light chain. There are just kind of a few things that are a little more obscure. So when we're talking about pigment, there was chlorophyll A and chlorophyll B, right? Do you guys remember that slide that was talking about it? And they're very similar. They're mostly the same structure, except chlorophyll A will have like a CH3 in one of the spots, and chlorophyll B will have a CHO in the same spot. But it's what makes the chlorophylls different. But both chlorophylls, chlorophyll A and B, will be in P680, and they'll be in P700. However, They absorb different wavelengths of light. That's the main difference between chlorophyll A and chlorophyll B.
And carotenoids are in both of them as well, right?
Mm-hmm.
Absorb
Do
different.
we need to know the structures of chlorophyll A and B?
No.
Absorb different.
They absorb different wavelengths.
Okay. So.
Yeah.
So it's just kind of like P700 has more, for example, chlorophyll B, and that's why it absorbs primarily at 700. And then P680 would have more chlorophyll of A.
So, okay, so the different pigments, the different amounts of pigments, different concentrations of pigments in photosystem 700 and photosystem 680, or P700 and P680, that will make up for the difference of why they absorb at different wavelengths. But I don't know if it's because it has more B or if it's because it has more A. Yeah, but it would be because of that different concentration. Alrighty, so another thing, paper chromatography. You guys know what that is? No? So the main thing that's important about paper chromatography is it's used to separate pigments. So it's how you can tell pigments will have like a certain concentration of chlorophyll A and chlorophyll B, and you can separate which pigment is which based on a paper chromatography experiment.
So it's just like a scientific method?
So what it is, it's a experiment that you. Okay, talk to me after. I don't want to. It's in the book. Yeah. I don't really want to get into the. I can explain it to you after class, but I don't want to confuse people and explain it up here. Unless everyone really wants me to, but I don't think people do. Yeah. Yeah. Okay. So let me reiterate. Paper chromatography is used to separate pigments. So the pigments, there are a few different types of pigments in each of the photosystems, in each of the thylakoid membranes, and you can separate those and tell like which pigment is the most by using paper chromatography. Okay. So last thing about light chain, then I promise we're done. Do you guys remember that slide in class where it was showing like I can't remember exactly how it went, but there was red up here, it was being highly absorbed, and then it went down. Green was very low, not very much green was being absorbed. Blue was high again. So when green is low, what can you assume about how much oxygen it will be produced? No.
Very little.
Very little, because it's all the light's being reflected, so we're not exciting this electron, so we're not hydrolyzing water. Yeah.
It's absorbing a lot of red and blue.
Right. So we're absorbing this light. It's exciting the electrons. That hydrolyzes this water. Produces oxygen.
That's a lot, right?
A lot. So red and blue would be a lot. Green would be a little.
Okay, that's what.
Oh, is that what you said?
Yeah.
Okay, I'm so sorry. I misheard you. Yeah, so red, green, blue. Okay.
Can you say that again? So when it's the green is low, you are not hydrolyzed with oxygen.
Yeah, so, okay, so let's talk about what's going on. Red. Okay. So, basically, red light absorbs really, really well. So, all of this light will go into P680 to produce this electron, which then goes through the whole chain, right? And when we absorb this light and excite this electron, something has to replace it. It's replaced with an electron from water. There's this hydrolysis reaction going on, and it produces oxygen. So with red light, we're going to produce a ton of oxygen because it's absorbed very effectively. However, green light is ineffective, and it will be reflected. So we won't receive all of this light, and this whole process can never occur. So we don't hydrolyze water, so we don't create a lot of oxygen. Okay, did that make sense?
Yeah, but so like overall, you are creating a lot of oxygen just with all the different wavelengths, but green specifically.
Yeah. Yeah, so I'm talking about if we break it down specifically where oxygen is coming from colors.
So then there's a lot of red and blue like wavelengths in the world, right? Because we have a lot of oxygen.
Okay. Light cycle is over, guys. I feel like it deserves a round of applause. There we go. Yeah. So now, can I erase this?
You erase it.
Okay, good. I'm so sick of it. So. Okay, so let's finish this off and talk about a few more things in our remaining time. So the second thing that's happening, excuse me, we have our dark cycle going on. Now guys, you really don't need to know a whole lot about dark cycle. He gave that slide in class, and he doesn't want you to know the ins and outs of it. It's not worth your time to memorize. Yeah?
Just quick questions. We've had multiple choice for all our past tests, but from some other students I've talked to in other bio classes, they had to like draw out.
Nope, multiple choice.
Okay.
Yeah, no, it makes me really happy as a king too. I don't grade it. Okay, so let's talk about who knows what carbon fixation means.
Making carbon.
Okay, yeah. So as animals, we can't create carbon. We have to take it in from some external source. We take it in through our food, but plants are cool because they can that carbon dioxide that they absorb from the environment, they can use to make into glucose and sucrose. Okay, so carbon fixation would be like a carbon dioxide molecule will bind to an organic compound. So a good way to remember it is just we have carbon dioxide, and it's bound to an organic compound, which would be glucose. And then glucose can then go make energy. All right? This is carbon fixation. So there's part of photosynthesis is creating this. We have this intake of carbon dioxide, not we, the plants do, and there's this cycle going around. And basically, what happens is we end up with something that has 5 carbons. These circles will indicate carbon. So I actually want to do this instead. So, and this will be called ribulose bisphosphate. And so, again, it's kind of like what came first. We don't know if it was this carbon dioxide that somehow was made into ribulose bisphosphate, or if it was this that combined with carbon dioxide. So don't worry about it too much. But basically what's happening is there's this enzyme called rubisco, and it's this huge enzyme, and it will take this carbon from carbon dioxide, it will put it on to ribulose bisphosphate. So now we have 6 carbons, and then it will cut it in half. And when it cuts it in half, you get 3PG, or you get PGA, which will go into 3PG, or G3P, sorry. And this can then go to glucose. But the remaining carbon will circle back around here to make ribulose bisphosphate, which can then, it goes in this circle. Okay, I don't think I explained that very well. So what questions do you guys have?
If you have questions, there's like a really good video, I think it's by Ted, and just look up Calvin Cycle. It's like a kid's animation, but it's so good. Like,
Okay.
would totally explain it.
Cool. So yeah. What else? What other questions do we have about this? What is this called? Oh, she said TED.
It's just like a TED animation. You search Calvin cycle by TED Talks. I don't know.
Where does it happen?
The stroma. Or no, yeah.
Yeah.
What are the inputs and outputs?
What are the inputs and outputs? Your big input would be carbon dioxide. Your big output is G3P, which is made into glucose. Those are the main things you need to know. Sorry?
You didn't talk about like blood respiration or anything like that in class. So you don't even know about like cancer and people.
You don't need to know about what?
Plants and people coming.
Okay, so this is my clarification.
Yeah.
There's 5 carbons in that, and there's the 1 carbon. Then it goes to the PGA and the G3P into the glucose.
Yep.
So how many carbons are being taken off of those steps?
So you'll split it into 2 right here. So PGA will have 3 carbons. And G3P, I believe it also has 3 carbons. But I think the rest will go here to make ribulose bisphosphate, and it will combine with other carbon sources along the way to get these 2 extra carbons. I don't know exactly what carbon sources it combines with though.
There are 6 other of these occurring at the same time.
Oh yeah.
And practically, basically what happens is they all give, I think it's 1, and then they come back to have those 5. It's kind of confusing, but they each give 1 to start, so the 6 give 1 to start one of the cycles again, if that makes any sense. Watch the video.
Yeah. Yeah, so unless you want to be a botanist, just know this. Okay, what else do I want to talk about with this? Oh, so one thing about Rubisco, so someone mentioned it already. It's huge. It's like this massive enzyme, and because it's massive, it's very slow. So we have to have, the plants have to have a lot of Rubisco so this reaction can happen because it's such a slow enzyme. Okay. All right. So just kind of a few last minute things. So we have this light independent reaction and a light dependent reaction. And how are they related? Sorry? Good.
So one's output is another's input.
Uh-huh.
And so then as they both work, then they can continue to feed off each other.
Yeah. So the light dependent reaction, it produced ATP, right? And that ATP is the energy source that allows the start cycle to happen, so it allows glucose to be produced. So if our ATP that we produced in our light cycle is going to make glucose, what does that mean? It means we need mitochondria. Okay? So this is a point that he made very clear in class. Plants have both chloroplasts and mitochondria because they're both creating ATP, but they're using it for different reasons. The ATP associated with the light cycle and just photosynthesis in general is used to make glucose, which is the nourishment for the plant. And the ATP generated from the mitochondria is used as energy for the plant to sustain life. Okay? Okay. This again was talked about in class. Could you ever grow a plant in the dark?
Yes.
Yes.
What would you need in order to do that?
Water.
Yeah, you need water and glucose, and you can grow a plant in the dark. Because the whole purpose of our light-dependent, light-independent reaction was to produce glucose. So if we just add that glucose externally, we can still have our plant grow and sustain life.
With the chloroplast mitochondria thing, so you mentioned that mitochondria produces ATP for the plant to sustain life, and why does it need the glucose?
It needs the glucose because that's how glycolysis happens.
Right.
So So that's the plants, as I said, they can make their own food, basically. They make their own glucose, where we can't do that. We need glucose for food.
So the light reaction produces
Uh-huh.
and you said that it can be like kind of fuels and happen cycle. So where is ATP?
You know, I'm not sure.
Just know that
Yeah. Okay. So the last two things I wanted to talk about, and this was mentioned in class a few times, catabolic and anabolic. What do they mean? Good. So, yeah, so exactly what she said. For some reason, it always makes more sense when the person up front says it again.
Yeah,
so.
I wrote
Okay.
it, yeah.
So, catabolic is a breakdown of large molecules to release chemical energy. And so, like glycolysis is an example of this. We have our big glucose molecule, and we break it down, and we create energy, right? So, anabolic, an anabolic reaction would be one that synthesizes molecules to store energy. They're opposites. So, photosynthesis is an example of an anabolistic, I think I made that word up, process. It is using resources to make a big glucose molecule which stores energy.
Can you say the definition of anabolic one time?
Uh-huh. It synthesizes molecules to store energy.
So it synthesizes to store energy, and then catabolic is just to break it down and just go get energy.
Yeah. Yeah. So one's a storage of energy, one is utilization of energy.
Would you recommend that we understand what the autotropes and other trope things are?
Nope. Nope.
Nope. And nothing, okay, even they have to do with photosynthesis.
You know, if you really want to, go for it.
Okay.
Yeah. So that's all I have prepared. If you guys have questions, I can stay after and answer them. Sorry, this was my really weak unit, so next review will be better.
Good job.